In this presentation, we are going to continue with tips on genetics. In the previous presentation, we saw how to understand questions and identify alleles and genotypes. Here we are going to continue with tips on how to identify the types of inheritances in a question. This presentation will not be very useful to a student who has not yet studied the chapter. Correct identification of the type of inheritance is crucial to getting your question right. It is by understanding the type of inheritance that you will know how to represent the alleles and the genotypes, and also how to interpret phenotypes from the genotypes in the offspring. Let us look at inheritance with dominance, which lays the foundation for understanding other types of inheritance. Inheritance with dominance is Mendelian inheritance. Recall that one of Mendel's laws is a law of dominance. To identify that you are dealing with dominance, note that there will be two contrasting phenotypes which are versions of a given character, and one of them, the dominant phenotype, appears in the F1 when crossing pure breeds, while the other phenotype, the recessive phenotype, disappears in the F1 and reappears in the F2. In dominance, alleles are represented with upper and lowercase of the same letter. The uppercase for the dominant allele and the lowercase for the recessive allele. A monohybrid cross with pure breeds gives the F1 ratio of 1, that is, all the offspring show the dominant phenotype. When the F1 individuals are self-crossed, the F2 appear in the ratio of 3 dominant phenotypes is to 1 recessive phenotype. Recall that the test cross aims at identifying the genotype of an individual showing the dominant phenotype. That is, whether the individual is homozygous dominant or heterozygous. In the monohybrid test cross, the homozygous dominant individual will give offspring with all dominant phenotype, while the heterozygote individual will give dominant and recessive offspring in the ratio of 1:1. In a dihybrid cross with pure breeds, that is, crossing a double homozygous dominant and a double homozygous recessive, the F1 individuals will all show the dominant phenotype. When the F1 are self-crossed, the F2 appear in the ratio of 9:3:3:1. In a dihybrid test cross with dominance, there are 3 possibilities. First, the double dominant phenotypes are double homozygous. Here, all the F1 will show the double dominant phenotype, as shown on the example. Second, the double dominant phenotypes are heterozygous. The F1 phenotypic ratios will be 1:1:1:1. See the example. What is important here is to keep the ratio. Third, the double dominant phenotypes have 1 heterozygous locus and 1 homozygous locus. The offspring come in the ratio 1:1. For the heterozygous loci, the respective recessive characters appear in the F1. See the example. Let us look at the inheritance with incomplete dominance and codominance comparatively. In both of the inheritances, there are 3 phenotypes. 2 parental phenotypes for the homozygous and 1 for the heterozygote. What makes the difference is that in incomplete dominance, the parental phenotypes all appear in the F1 and reappear in the F2. They blend in the heterozygote to give a new phenotype. While in codominance, the parental phenotype all appear in the F1, such that F1 individuals show both parental phenotypes. Very importantly, for both we use different uppercase letters to represent the alleles, unlike in dominance since no allele dominates. The F2 phenotypic ratio is 1:2, is:1 instead of 3, is:1 in dominance. The ratio 3 is split because the homozygous dominant has a different phenotype from the heterozygote. Incomplete dominance is common in plants, for example, flower color inheritance in the snapdragon flower. Codominance is common in animals, for example, roan color in cattle, which is a mixture of red or black and white obtained by crossing red or black and white cattle. Others include the A and B alleles of ABO blood group and the A and S alleles in sickle cell. For inheritance with linkage, the genes basically show inheritance with dominance, but deviate from Mendel's laws because they do not show independent assortment. It is observed with dihybrid crosses and can easily be confused with Mendel's dihybrid cross. To identify linkage, look at the test cross ratio for F1 individuals produced from pure breeds. That is, the double heterozygotes, which will show a large number, X is to 1, is to 1 is to the same large number X, instead of 1 is to 1 is to 1 is to 1 in dominance. To represent the alleles, use upper and lower cases of the same letters to represent alleles, just like with dominance. However, since the genes are occurring on the same chromosome, that is, they are linked, alleles are written on bars to represent genotypes. The bars representing the chromosome. In epistasis, you will be given 2 genes conditioning the appearance of a character. The genes basically show dominance, but one modifies the expression of the other. The character will be given 3 phenotypes, for example, 3 different colors for fur color in mice. Use upper and lower of the same letters to represent alleles for the gene that determine character, that is, the hypostatic gene, and for the epistatic or modifier gene. Only the interpretation will change because alleles at 2 loci will be interpreted as one phenotype. To identify that a question epistasis, the F2 ratios will be a modification of the Mendelian dihybrid 9 is to 3 is 1, that is, the ratios sum up to 16. For example, in recessive epistasis, for example, the inheritance of fur color in mice gives the ratio of 9 is to 4 is to 1. What we aim to do in this presentation is to look for tips to understand questions. Kindly read each subtopic, and these tips will be useful to you. We have seen the various ratios for the inheritances. Try out your understanding by identifying the types of inheritance and crosses corresponding to the following ratios. Note that one ratio can represent several crosses. Here, let us look at the various stages in representing crosses in your exercises. First and foremost, identify the character in the inheritance. This carries no mark B, will help you to know how to represent the alleles in the genotype. For example, you can state clearly as character, seed color with dominance. In the next step, state the alleles. Each stated allele takes a mark. Next, state the parental phenotypes. This step takes a mark. Then the parental genotypes, which also takes a mark. From the genotypes, separate the alleles or permute them to form gametes and state their ratios. Gametes take a mark and ratios take another mark. In the next step, cross the gametes to form the F1 genotypes and state their ratios by multiplying the ratios of the gametes. Genotypes take a mark and another mark goes for ratios. Finally, write the F1 phenotypes and their ratios, which take another mark. We have come to the end of the second part of this presentation on tips on genetics. In the next presentation, we are going to take an example to present how to apply these tips. Please subscribe to this channel, like, and share so as to get notifications of new presentations. Thank you.